Good morning. So welcome everyone today under the circumstances. Thank you all for coming. We appreciate you coming for the 17th Greenebaum Lecture Series. I just want to start off and say a couple of words about Greenebaum Lecture and then hand over to the next speaker. So the Greenebaum Lecture has been a key scientific piece of Institute of Human Virology and the University of School of Medicine for 20 years. This is the 17th lecture where we have honored scientists, leaders, clinicians, and public health experts at various arenas over the last 20 years. We are extremely thankful for a generous donation from Stuart Greenebaum and now from the Greenebaum family for the conduct of this particular lecture. For those of you who remember, in the past Greenebaum lectures, Stuart Greenebaum used to come to all of them. He used to sit here and listen to it, and often have lunch with the speaker. And he was a very integral part of this conference and lecture series. Greenebaum family is not stranger for us. They have worked closely with the University of Maryland and the Cancer Center and have given a lot of philanthropic efforts. And the family members used to be part of the celebrations. And I really want to take the opportunity to express sincere gratitude. The Greenebaum family unfortunately couldn't be here, to this Michael Greenebaum and the family for this generous donation. We have a great speaker today, Dr. Jeanne Marrazzo, the director of NIAID, who will be introduced later. And at this point in time, I also want to thank the people at IHV who helped us organize this. And it's particularly all the staff members and faculty who helped us, in name particularly Danny O and Lana Rosenthal, who were instrumental in making sure all the logistics have been done. They couldn't do anything about the gas leak, but everything else was done fine. I also want to remind everybody that there will be lunch will be served after this meeting. So please stay for lunch and make sure that this is going to be on the right side as usual. So I'm going to hand over to our Dean, Mark Gladwin, to say a few words.
Thank you so much, Dr. Kodalil. And I just want to extend a warm welcome to Dr. Marrazzo. We really appreciate you being here with us. We're sort of proud to think of ourselves as a branch of NIAID. Over the last decade, we've received almost $1 billion in funding from NIAID. And then the last year, NIAID represented 24% of our funding portfolio. So we appreciate all the support that we've received for innovative science. I would just say a few comments about IHV. Part of my job as the dean is to be the cheerleader, but I spent a lot of time this summer with our faculty leaders of IHV in Nigeria, and it was just an incredible experience to see the IHV Nigeria site at Abuja, to travel to Lagos. Over that time, I learned that really over the last 20 years, there's been almost a $1 billion investment from the federal government through the University of Maryland, focused on global health of containing the HIV pandemic. And in that time, our faculty and the partnerships we built with Nigeria were responsible for screening 30 million people and treating 8 million people, and almost 25% of the capacity for current HIV treatment in the continent of Africa is through this long-standing partnership with the University of Maryland and Nigeria and other countries in Africa. So we're very proud of that global health legacy here. And obviously, this couldn't have been possible without the support of the CDC and the NIH, and we really appreciate that. I also do want to thank Jeanne for organizing this. I want to thank the Greenebaums, who have been incredible supporters of the University of Maryland, and thank all of you as our faculty for the important work you do. And with that, I'll ask Dr. Rosenthal to come up here and provide a formal invitation. Thank you.
Thank you, Dean Gladwin. I'm excited to introduce our speaker today, Dr. Marrazzo, who's the director of the National Institute of Allergy and Infectious Diseases at the U.S. National Institutes of Health, where she oversees a $6.6 billion budget that supports research to advance the understanding, diagnosis, treatment, and prevention of infectious, immunologic, and allergic diseases. Prior to her position at NIAID, Dr. Marrazzo was the C. Glenn Cobb's Endowed Chair and director of the Division of Infectious Diseases at the University of Alabama at Birmingham School of Medicine. And before that, she served as faculty at University of Washington for over 2 decades. Dr. Marrazzo is internationally recognized for her research and education efforts in the field of sexually transmitted infections, especially as they affect women's health. Dr. Marrazzo's research in discovery and implementation science has focused on the human microbiome, specifically as it relates to female reproductive tract infections and hormonal contraception, prevention of HIV infection using biomedical interventions, including PrEP and microbicides, and the pathogenesis and management of bacterial vaginosis, sexually transmitted diseases in people with HIV, and management of antibiotic resistance in gonorrhea. She is author, co-author, or editor of more than 250 scientific publications and a member of the National Academy of Medicine. Over her career, Dr. Marrazzo has also focused on the development and support of trainees, the advancement of underrepresented minorities in medicine and leadership, and promoting meaningful dialogue with communities and patient care. She has been a leading voice in communicating science during the COVID-19 pandemic. Dr. Marrazzo was the recipient of the American Sexually Transmitted Diseases Association's Distinguished Career Award, the highest recognition of contributions to research and mentoring in the field. She is a fellow of the American College of Physicians and the Infectious Diseases Society of America, for which she served as treasurer from 2021 to 2023. She has had leadership roles in the NIH HIV Prevention Trials Network and the Infectious Diseases Clinical Research Consortium, and has chaired the American Board of Internal Medicine Council and the ABIM Infectious Diseases Specialty Board. And though it's hard to imagine that she has much free time, she is also an avid birder. So it goes without saying that we are incredibly honored to welcome Dr. Jeanne Marrazzo as the 17th speaker for the IHV Annual Greenebaum Lecture. Welcome, Dr. Marrazzo.
Well, it's a huge honor to be here. I've worked with so many of you over the years in varying capacities, and so to get to see you all in person, especially on a day where you had barriers to actually get to buildings with power, really means a lot. So I really want to thank Dr. Codalil for the invitation, Dr. Rosenthal for that lovely introduction, Dr. Gladwin, Dean Gladwin for welcoming me, and I'm excited to talk to you. I've been at NIAID now just a little over a year. I arrived in Bethesda on October 15th. So you really are getting a very fresh look. And I will say it's an evolving look because the learning curve is pretty steep. And I have some thoughts, but they are not as informed as I'll be in another year. So just to put that in mind. So what I'm going to do today is to provide, in part because I know most of you don't probably really know that much about me, despite that nice introduction. Those introductions only give you a little bit of a picture of your CV, right? And how I got to where I am and why I took this job. And then I'm going to highlight some NIAID activities that I think you will find very interesting. I'll talk about the budget. I'll talk about some challenges with our budget. I'll talk about some single discoveries and case studies that highlight how people who do the work you do change the world. And that's really what I want to thank you for and to talk about. Talk a little bit about future strategies, aspirations, and challenges. And again, really want to thank you for all you do to advance our collective mission and vision. So when I decided I wanted to go into the infectious disease field, and I promise there are only 2 slides on my background, so it's not going to be that boring, maybe 3. I was really interested in infectious diseases for the same reason that many of you in this room and many of the people who started this institute were interested in. I trained in New Haven, Connecticut in the late 80s, early 90s. And at that time, New Haven had a HIV epidemic that was pretty similar to many urban HIV epidemics. So lots of gay men, lots of injection drug users, and lots of people who acquired it through heterosexual relationships, particularly young Black women. In that regard, it really resembled the epidemic that many people wanted to go to study in sub-Saharan Africa. And I know that Slim and Kareem Abdul were on your wall. They've been here before and gotten your award. And they were, of course, recently just won the Lasker Prize for documenting for one of the first instances, how high the prevalence of HIV incidence was in those young women in sub-Saharan Africa. People really had not been focused on that. So I knew that I really wanted to do HIV, but if you did HIV in those days, as Henry knows well, and many others, you really learned pretty quickly that you had to be an advocate for your patients, right? The stigma is bad now in many places around HIV. Having been in the Deep South, I can tell you that it's alive and well there and still killing people. But in the early days, you really had to learn how to advocate for your patients because they didn't have a lot of people standing up for them. I was very fortunate because I was trained by people who were not just strong researchers, but strong educators. And they also had a rare sense of a commitment to public health and the impact that the work that they were doing. I was also very interested in women's health. Again, that epidemic in New Haven really brought that home. So I was really fortunate to have terrific role models. And I will point out that one of my very first role models was my mom. So my mom is shown there. She's a nurse. She's a very healthy 97 right now. She still drives, I'm sorry to say. This is her she's born in the US, but she went to Italy with my father in the 50s, ended up working in a US Army hospital in Livorno, which was interesting where I was born. But they came back to the US and she worked as a nurse in Scranton for her whole career. And in the mid 80s, as I was kind of getting ready, I guess I was in college and then in medical school at that time, she was an oncology nurse and she started taking care of a lot of young men who were returning to Scranton to die. With AIDS because they were coming home from New York City, they were coming home from Philadelphia, they were coming home from Pittsburgh to their families to die. And of course, oncology was where palliative care was done. It was also, remember, a huge field in the early days of the HIV pandemic because of Kaposi's sarcoma being one of the earliest conditions that was detected in these men in particular. And I will never forget her telling me that there were plenty of doctors and nurses who refused to see the patients. And I know that is not news, but in this day and age, it really brings home how, again, the stigma affected compassionate care, particularly at the end of life for these people. And my mom, who is not really one to make waves, was a really fierce advocate for these patients in her own quiet way. So a huge role model, really important, and informed a lot of my early commitment, I would say, to the field. So second of the last two slides that are going to talk about me, went to University of Washington, almost came to Baltimore, was really this close to come to training here and ended up going to the West Coast, where, as Elena said, I was spent most of my career with a focus on those things at the bottom, and then went to the University of Alabama at Birmingham in 2016. And it's worth saying I went there because of the people who were leaders there. So the dean who recruited me is Dr. Selwyn Vickers. Selwyn's now the president of Sloan Kettering Memorial. He's a cancer surgeon who trained here in Baltimore, actually. His grandparents were sharecroppers. His father couldn't get his PhD until he was well into his 50s. And incredible role model, incredible realization of how really strong leadership can change the trajectory for so many people in training and who are in their career development. So, and then last fall, I came to the NIH to begin my tenure as the 6th NIAID director, and that's Dr. Monica Bertagnoli, my boss, swearing me in with my wife, Jo Hoffman, who's been through a lot with me and been very supportive. So for those of you who don't know NIAID, what do we do? Many of you in this room have, it sounds like, been supported by us for many, many years. But what we do is to support and conduct biomedical research to better understand and treat infectious, immunologic, and allergic diseases. But what I want to point out here is, and I want to also really emphasize that it's not just HIV, of course, that we focus on, but we focus on the whole other world of infections. So there are, like 235 other pathogens. But very important to this group, a very, very big focus of our work is on HIV. And if you look at our budget, about 30% actually funds directly HIV-related research. And that's largely therapeutic and vaccine development, but also community outreach. So this is sort of who we are and what we do. But my point of this is that and I did a variation of this talk at Infectious Disease Week last weekend, and that's a lot of clinicians. And what I wanted to sort of convey to people is that we affect not just the people who write our grants, but we develop the tools that people use to affect change with communities and patients. So these are some examples of things that started in either our laboratories or with grants to people who then move them forward to make huge interventions that got to the patient or the community level. So I think one of the best examples, of course, is Barney Graham's work on the RSVF protein, locking that in the pre-fusion state so that you could create an immunogen that was stable and actually led to the development of the RSV vaccine. It's because of that advance that we were able, and they were able, to develop a SARS-CoV-2 vaccine so quickly because it was essentially the same technology. If you're an allergist, you know that the peanut allergy world has changed a lot, largely through studies done through NIAID. So the LEAP trial was the study that showed pretty definitively that early exposure to peanut allergy is really good. You don't want kids to wait to get exposed to peanuts, so they should be getting peanut exposure pretty early. And then a couple of other examples there, which I won't go into, but you can take a look at. So how do we do all this? Well, we have been very generously funded for the last several decades. You can see that from 2000 to 2024, our budget has advanced dramatically, largely due to excellent advocacy from my predecessor in our legislative branch and the team, but also due to the science that you do, right? We don't have any trouble representing how incredible the work that we fund is, because you do great science and you do great work. I will point out, though, that you can see that our budget, including the president's budget for 2025, is essentially flat. So we have not had a flat budget for the last 2 decades. That sort of pseudo dip there is actually because some funds were rerouted. So that really isn't a dip. It actually continued to go up. So we have been extraordinarily fortunate. And in this year, it looks like we're going to do okay, although we don't know what's going to happen, of course, in the next several weeks. We're in a continuing resolution until December 8th or so. But the president's budget has requested a small increase for us, about $20 million. And you may remember that there are 3 budgets that are proposed, if you remember your civics class. There's the House, the Senate, and the president, right? So the president's budget is what I showed here. The House budget, when I came to NIAID last year, the House was proposing that NIAID only institute would be cut by 23.9%. So that was well over $1 billion. And there was a lot behind that impulse. It was not a friendly impulse. And we ended up not seeing that. And I consider it a gigantic victory that this year the House did not propose a cut in any of our budgets, which I think is amazing. They did propose splitting us into 2 institutes, the National Institute on Infectious Diseases and the National Institute on the Immune System and Arthritis. Interestingly enough, even apparently they didn't realize that there's another institute that focuses on musculoskeletal disorders. I know I shouldn't say things like that now that I'm a government employee. So again, these are the kinds of things when people ask how I spend my time. Doing stuff like this is a super fun 1%. Much of the time, as those of you who do administration know, is spent making sure that we have the resources to get to the people who deserve it and who need it. So I'm just going to show you what the budget looks like and where we stand. These are the top institutes, top funded institutes at the NIH, and you can see that the National Cancer Institute is just above us at a little above 7.2. So in 2024, our budget, as was stated, is around 6.56. The House proposed not just a decrease, but a slight increase. Again, I think that's a really strong vote of support. And then the Senate proposed a 2% increase. So we'll see what happens. We are beholden to earmarks. And if you don't know what those are areas where we have to spend funding in a particular area. So for example, we have to spend a lot of money on tick-borne diseases, whether we want to or not, because those are funds that have been allocated through the Congress for that purpose. We have to spend money on biocontainment laboratories. We have to spend money on a couple of other things, that influenza, actually. So it's worth noting that. And because you are, as Dean Gladwin said, such an incredible global health force in your research, I wanted to make sure I talked a little bit about our investment in global health. A lot of people don't realize that we spend about 10% of our budget on global health research. And we do that either with direct grants that go to these countries through some of the bilateral agreements that I'm going to show you in the next slide, or more commonly through the partnerships that we fund to you, and then you work with the people in these countries. And so it's quite a lot of money. I was in South Africa in August to visit several of our sites, and at a reception, the consul general actually mentioned that in the last several decades in South Africa, we've invested over $1 billion. So it's quite a lot of money with, I think, really amazing results. And you can see the top 10 countries funded there. And we have formal bilateral programs, which means that we really have some infrastructure, some investments. These are the places that I hope to visit in my sort of early years. I've gone to South Africa. I'm going to Brazil next month. So these are very, very strong relationships that we have cultivated with lots of scientists growing up through these collaborations. And that's really what you want to do, right? You want to sort of foster a coalition of investigators across the globe through which you can not just access and work with local samples, local communities, but create professional development for these countries. So what am I going to talk about in terms of impact and opportunities in ID research? And I do hope to have some time for questions. You can tell me if I'm talking too fast. Sometimes I get a little anaerobic in my speech. But mostly what I want to talk about are viruses, because you are the Institute of Virology. I'm not going to talk a ton about HIV. I think you probably hear a lot about HIV, but we can certainly talk about that. It's my passion. It's where I feel probably most comfortable talking about things. But we can come back to that. I think the field is really interesting right now in terms of chemoprophylaxis and opportunities there. But there's so much other stuff going on in the world right now, and that's really where I'm going to go. So when you look at what we have for opportunities, and again, I talked to medical students and residents who attended ID Week this week to sort of try to help them think about why ID is such a great field and what the opportunities are. You can see that we are truly not on top of most infectious diseases. Either they're escaping and re-emerging, or we're continuing to see new challenges. And that's not just outside the United States in areas we traditionally think of as the global arena, but also in the United States, right? We're now you have Maryland, you have dengue in Maryland, you have malaria in Maryland. You have lots of these things that are coming up. And one of the major concerns is zoonotic spillover, right? As we destroy habitat across the globe and destroy ecosystems. And it's estimated that about 75% of these emerging pathogens are zoonotic. The other challenge with that, of course, is climate change. So climate change is accelerating disease emergence and spread. And I just want to spend a couple of seconds talking about that, because you really can't disentangle these two things as they evolve. So you may recall last year smashed records for the world's hottest year by a large margin. And why is that important in infectious diseases? The most obvious thing is that vector ranges change. And so not only are you creating that conditions that are more promotive for things like mosquitoes to thrive, but you're also extending their range because of the change in temperature. And I'm going to show you a nice graphic about that in a second. There are also more frequent extreme climatic events, and floods, as you have seen, can cause surges of infections, particularly enteric infections. People's water supply is really damaged. And also these things trash the infrastructure. So one thing I will remember about mention about HIV, I've been getting a lot of questions from people about lenacapavir working so well, given every 6 months. Why do we even need to pursue an HIV vaccine, right? Well, to administer lenacapavir every 6 months, you need a pretty stable, intact infrastructure with well-trained staff who can actually do the subcutaneous injections, 2 injections in the belly, not something that you can just have anybody go out and do and give an intramuscular injection. So think about Rwanda, right, in Marburg. that could have been taken down in terms of the healthcare system if Rwanda was not so well prepared and didn't have such a robust healthcare system. So there are just so many things that can threaten the fragility of the infrastructure that I really worry about not having durable individual immunity for HIV. So that's one of my big arguments. And then, of course, climate change really brings home the concept of One Health, right? When people migrate, animals migrate, livestock migrate, and we're seeing that, of course, not necessarily with climate change, but certainly with H5N1, which I'll talk about in a second. So this is an animated graphic, and what you're seeing here is, I think, kind of chilling and also, again, being borne out. But this is a model that looks at the expansion of Aedes aegypti mosquitoes in the US and China, two of the areas, of course, that we are most concerned about. You can see the rest of the world is down there. But this is over the period of 100 years, 1950 to 2050. And I think you can't really argue that this isn't happening. We're seeing it happening in real life as these infections continue to move. This is being accompanied, of course, by parasitic drug resistance. So we're now seeing Aedes stevensii in Ethiopia, which is very, very resistant to antimalarials. So it is not a good combination of events. So I'll say a few words about malaria. It's not all doom and gloom. And I think, in fact, that if we think in the next 5 to 10 years of infections that we are going to see profound impact in, I have the top 3 for consideration: HIV, because of what I just said, and malaria is definitely one. We'll come back to the third. So malaria, of course, as you know, being the Global Health Institute, is still a major problem. We have 2 nice vaccines, the R21 and RTSS. Problem is they're not super effective, right? The most you're going to get out of them is maybe a 30 to 40% efficacy. And, you know, we need to sort of remember that. I think it's exciting, but we still need new tools, and we are not continuing to affect declines in the numbers. In fact, there's a slight increase there in 2020, perhaps around all of the pandemic disruptions, and also, as I said, accompanied by the specter of resistance. So just a couple of examples, again, about how things come out of basic science, in this case, the intramural program at NIAID, and actually are very likely to change the disease trajectories globally. So some of you may have heard about the monoclonal antibodies that have been developed to prevent malaria infection. And one of the first was this monoclonal that targets the circumsporozoite protein. That's the prehepatic phase of the pathogen. So the idea is you try to take these pathogens out before they even establish the hepatic phase, and thus can't disseminate to get to the lungs, for example, which is one of the big problems that we see. And so the first antibody is this one, and it was really a proof of concept study that was published in the New England Journal in 2020 in Malian adults, and showed that it was very safe and highly protective. And you can see the people who did not get the antibody had a likelihood of 78.2% infection. So malaria, of course, is endemic, and you're going to get it if you're out there. But if you got either doses of these, there's a high dose and a lower dose, you had a pretty remarkable efficacy, and this was over 6 months. So durability was pretty good. Why is that important? Because that could get you through a season, right? When you think about seasonal malaria, you want to think about seasonal interventions. So what do you do when you find an intervention that looks good? You try to make it better. And when you're talking about monoclonal antibodies, what that means is you try to make them more potent and more durable. And indeed, this is the second antibody that is 2 to 3 times more potent, the L9LS, again, targeting this very conserved region in the circumsporozoite. This is the first, this was a human infection model, and I know you are very familiar with those here, and you have some fantastic malaria investigators here. So this showed pretty definitively that 4 of 5 people were protected with subcutaneous injection, and that's important because subcu injection for ease of implementation in the field is a heck of a lot easier than intravenous injection, right? You don't want to have to set up an IV, and that's going to come back when we talk about the malaria vaccines. So that product, so that looks really good. That product was then taken into a study of Molly in children. And again, the beautiful thing about monoclonals is they're safe enough. You can give them to kids, right? And kids get the worst cases of malaria. They get cerebral malaria. Their mortality is higher. So it's really a high impact population. And this just shows you the cumulative incidence of the first infection by blood examination or the first clinical episode in these children in Mali. And this was published just this year in the New England Journal. And you can see that the efficacy was anywhere between 66 to 77%, depending on the endpoint, depending on the dose that you use. So you can start imagining that if you can wield these antibodies with a sub-Q injection in people most at risk, you could start to make some major population level impacts. And then if you couple that with a vaccine, think about what the possibilities could be. And I love this study, also published in Lancet Infectious Disease just in August, because this shows you the potential of the vaccine, but it also emphasizes how we can start doing studies once we know these things are safe and effective in the most vulnerable population. So this is a cool study, because what they did here was to enroll men and women, adults, but they enrolled women in particular who were intending to get pregnant in the next year. And I can't say how important that is, because that's a population who has been traditionally excluded from many of these studies, right? If you either can't be at risk for getting pregnant, you have to be not having sex, or you need to be on a highly effective hormonal birth control, and your partner theoretically needs to use a condom. I mean, it's completely insane and unrealistic and doesn't replicate any real world situation that I think most of us are familiar with. This study, in contrast, was very interested in getting women who they weren't pregnant when they started, but they could get pregnant. What did they see? They actually showed that they treated everybody presumptively to make sure they were clear of parasitemia, and then they randomized them either to a control or to 2 doses. And you can see that the vaccine efficacy was really significant. The graph here is shown specifically about the vaccine efficacy against malaria that occurred in pregnancy, because a substantial number of women did get pregnant. So very nice design, again, very inclusive, and trying to get the target population, because malaria in pregnancy, what happens? Stillbirth, mostly spontaneous abortion. There can also be a lot of pregnancy related morbidity and mortality for the mom. So not a good thing. The other thing I like about this is that women who received the vaccine conceived sooner than those on the placebo. So talk about a way to address vaccine hesitancy. It's like, if you want to get pregnant, get the vaccine. I don't think we can really say that yet, but I think it's fascinating. And the placental biology around this, there's a person whose name I should know, who's at NIAID, and Henry may know, but she's done placental biology for her whole career, and she's really looking carefully at what's going on here. So so they've got tissue from the women in this study, depending on what arm they were in. So the way to learn about this is going to be, I mean, what this is going to do to illuminate the field is very exciting. So I'm very excited about malaria. I think we have a chance now to do some work that is, again, growing out of some very incredible basic science, and again, needs people to continue to study it in the field. Dengue, of course, is just taking off incredibly. I was just in Lima last month at the HIV Research for Prevention meeting. And Lima, if you haven't been there, it's kind of a desert. It's very dry. It's on the coast, but it's between the ocean and the Pacific and the mountains. And they don't get a lot of rain. Really, when you're studying tropical medicine there, you usually need to go north to Iquitos, which is the mouth of the Amazon, to see dengue and malaria. Now they have local dengue in Lima, which is really surprising to people. And of course, we are seeing it, huge outbreaks in the Caribbean, and dramatically increasing across the globe. And the problem with dengue, of course, is it's not a good infection to get at all. I've had several friends who've gotten it and really were quite wiped out. But when you get it more than once, you increase your likelihood of getting infected with a different serotype and then getting severe dengue, which can cause a hemorrhagic fever. And that's a really potentially mortal condition. It's been traditionally very hard to develop a dengue vaccine. And you may remember the trial in the Philippines that was, I can't remember if it was stopped early, but it was a very a challenging one, because people were getting the vaccine and then getting infected again, and that was increasing their risk of severe dengue because they had antibody from their previous natural infection. So what you probably want to do is have a dengue vaccine that covers the main serotypes. And indeed, that's what the Butantan vaccine does. That was developed in partnership with our intramural program. And that has constructs that cover all the relevant antigens for dengue serotypes 1 through 4 and that's really what you want to do. How did that work? That was just published in the New England Journal this year. And again, incredibly encouraging, right? In an ongoing phase 3, double-blind trial in Brazil, which has a big problem with dengue. They're gonna follow these people for 5 years, but this is the 2-year data. And they showed that the overall efficacy was almost 80%. And critically, it was almost 90% in people with a history of exposure, showing that it didn't matter if you had been previously infected with these other serotypes. So that's a huge biological win. And I think the immunology of that and what that's going to do to allow us to figure this out and the pathophysiology of severe dengue is going to be terrific. It was also studied in people of all ages. It looked good, even in kids 2 to 6 years old. And then they weren't able to study the efficacy by serotype because they didn't have 3 and 4, but looked pretty good for 1 and 2, 90 and 70%. So again, another area where I think we can effect pretty impressive changes and control in the next couple of years. I'll just spend the next few minutes talking about pandemic prevention because we are the only institute at NIH that has the task of responding to public health emergencies. So that's what happened during COVID, during any pandemic or any public health emergency, whether it's an anthrax outbreak, whether it's coronavirus, whether it's H5N1, we really have to pivot quickly and use our infrastructure and our scientists to look at pandemic prevention. So we have a pretty nice pandemic preparedness plan that actually came out in the middle of the COVID pandemic. And the idea here is that there are many families of viruses that have pandemic potential. You can rank them all. You can look at their ability to transmit, to cause severe disease, whether we have medical countermeasures. There's no way you can have a vaccine or an antiviral or a monoclonal antibody on the shelf for all of them. It's just not feasible, particularly if you need to have clinical data to say it's okay to give this to people. So our approach has been to have something called a prototype and priority pathogen, where we characterize viral families that have pandemic concern, look at the one that's most likely to give us trouble, and then go ahead and go full bore on developing medical countermeasures for that. So that's what we have been doing. And the idea here is to shorten the timeline between outbreak onset and getting those countermeasures out to people. We have 3 major projects. Some of you may be part of this that I'll just mention. One is the Revamp project, and that's specifically tasked with developing vaccines and monoclonal antibodies. These are extramural projects, by the way, in addition to our VRC, which I'll talk about in a minute. We also have, this is an intramural project, the Premise Project, Danny Duack. He's got a very interesting program where he essentially does microbial and immune surveillance and epidemiology. So he collects plasma from all samples from all these places that have potential emerging viruses and characterizes the antibody response in people and is able to look at anticipatorily what might be coming out in those regions. And then, of course, we have an antiviral program for pandemics, which is tasked with developing antivirals. You work in virology, you know we have not had a huge amount of success in developing antiviral drugs, right? Even when you think about what we have for influenza, H5N1, it's okay, but you got to take it early. And once you get really severely ill, the impact is truly not great. If you've ever treated anybody with a viral pneumonitis in the ICU, you know that it's just, by that point, it's out of the range of what you can do with an antiviral. So I'll talk a little bit about H5N1. I was just at a meeting at the National Academy on Sunday that talked about this, and they are having a meeting today and tomorrow. They're going to work on a report. This is a really interesting politically, scientifically, epidemiologically, and immunologically. It's like watching a, I don't know, like a nerdy version of Succession, play out or something. It's been fascinating to watch the interagency response, to watch the criticism from experts in the scientific community, particularly Twitterati, and then to see what's happening in the last couple of days. And this slide's already out of date. This is just from my presentation on Sunday. We updated it yesterday. So I think you all know what's going on here. I don't need to belabor the point, but we're seeing outbreaks in humans that are from dairy contact, from infected milk, and also from poultry. And we just had now, for the first time, 4 cases reported in Washington State this weekend that were due to poultry exposure. The big outlier here, and you can see the epidemic outbreak curve down there that breaks down what those infections were from and when they occurred and the numbers. The case in Missouri is a real, I think, conundrum. There's a person in Missouri who had pretty significant pulmonary disease who presented to the hospital with gastrointestinal symptoms, ended up in the ICU because it progressed to pulmonary involvement. Patient was discharged before the test came home as positive for influenza. And then it was when it was realized it was not H1N1, it was typed as H5N1. That was after the patient went home. Patient did okay, was treated with oseltamivir, but there are still several people who are contacts out there who became ill. And we haven't been hearing about them because the CDC is still waiting on the serology. So the CDC is working on serology. We expect to hear what those results are next week. This week or next week. None of those people was tested at the time because nobody knew the patient had H5N1. So you start to see how challenging this is, and no contact with any animals or anything that might have suggested spread. So that's a little bit worrisome to me, to be honest. There have been multiple other species in this outbreak: alpacas, cats, house mice, and goats. And, when you look at the genetics of what these virus is doing here, you can see some changes from mammalian adaptation. So far, it hasn't adapted critically to bind to the human respiratory tract. That's really when we start to get very nervous. That's what might happen when it gets into pigs. I can't believe it's not already in pigs already, but that's the ultimate mixing bowl. So we'll see what happens. Thank you for mentioning that I'm a bird watcher. I am an avid birder. I really just like to be outside. But this is a picture of me actually doing shorebird surveillance. So I'll remind you that this virus has been in wild birds now for several years. And interestingly enough, when it first comes into bird populations, you can see pretty significant die-offs. You may have heard about the bald eagle populations dying off, seabird populations dying off. But one thing you really need to look at, and this is almost certainly why this outbreak started in Texas, almost certainly these were migrating birds that were part of that central flyway shown in blue there, that were going north from South America, where there's been a big outbreak affecting sea mammals, lots of sea mammals dying, and other birds, and then flying north, stopping over in Texas. Texas is a big place where birds fuel up to go north. They're going north to the tundra largely for breeding in the summer. So this is almost certainly how this started out: infected the dairy cows and spill over into the dairy cows, and then the dairy cows got it. I also show this because this surveillance is funded through us by something called the CEERS, the Centers for Excellence in Influenza Research and Response. And the CEERS don't just do studies in animal models. They are doing that to look at transmission and that kind of thing. But they also do active wild bird surveillance, which is what I'm doing there. And that bird that I'm about to hold is called a ruddy turnstone. It's shown on this slide. These birds literally just stop over in Delaware Bay because of the horseshoe crab stopping over. So they fatten up on those eggs and then they go north. Those birds that we are looking at there have a prevalence of influenza, which I know because I swabbed its cloaca, 27%, but they don't get sick. So part of what the SEER people are doing are trying to figure out why the birds have been really able to tolerate this infection so well. What are they doing? They don't get infected in the lungs. It's not, are they making antibodies? So it's a really active and exciting area of research. And if you don't do this work, you really can't predict when things are going to come forward. You may also detect new mutations that are going to be able to inform what happens when you see them in the human host. A couple of other things that the SEERs have done that I think are really exciting. They were among the first to look at the milk coming out of the cattle. They were able to show that you could transmit it in the ferret model, which is the big model for studying influenza, but really not very efficiently. So one of the things that the National Academy this weekend that really came up, and I think people feel comfortable saying, is that right now, at least, this is unlikely to be transmitted through the respiratory route, that it really probably requires direct contact with an infected surface, whether it's in the milking parlor, whether it's at the, at another person's eye. And so for right now, it's looking, I think, relatively reasonably self-contained. There is potential though, right? The alpha-2,3 sialic acid receptors are the avian receptors in their respiratory tract, but we also have them in our eye, and that's why people are getting conjunctivitis with this pathogen. If it switches to the alpha-2,6 sialic acid binding site, which is our epithelial binding site, that's when you really got to worry. It does have that potential, which is what we're showing here, but so far, we're fingers crossed. The other really cool and complicated thing about respiratory influenza infections is that they're not happening to us in a vacuum. So when you're born, you're part of a birth cohort who before the age of 5 gets exposed to an influenza virus. So you've all been imprinted as kids, and in successive generations, we get imprinted to different types of influenza virus. That's going to be in the background. And one of the cool things that Seema Lakwadala has done, she's at Emory now, was in Pittsburgh before she went down there. She has shown in the ferret model that if you infect ferrets with a strain of influenza, let them get better, and they do get better. They don't usually die. It's not a lethal infection in them. And then you expose them to highly pathogenic avian flu, they do better. So pre-existing immunity is almost certainly important here. It's one of the reasons people feel like getting vaccinated, even for the seasonal flu vaccine, although there's probably not direct overlap, might really make sense. And it's probably, again, the reason that we all know that young people didn't do so well in the 1918 influenza epidemic. So very exciting. Couple of words before I finish up with Mpox, because I do want to say a couple of things about that. I think I mentioned our vaccine research center, and I just want to point out, given your interest in vaccines in particular, that we have a long history of doing all the things that are shown here. And perhaps one of the most exciting and most relevant for this talk is that we have just started a phase 1 study of a stem cell-based nanoparticle vaccine for universal influenza. So remember, influenza virus has the H and hemagglutinin and neuraminidase. They change all the time. That's the HN. But the stem component is one of the most common parts. So this stem cell nanoprotein vaccine should be a very interesting to keep your eye on. We do need we need a universal flu vaccine. Couple words about Mpox because it's so profoundly affecting people in the Democratic Republic of Congo. I won't go through this timeline, but you know that it's now spread beyond that country. And we've had some disappointing, I would say, results and some encouraging results in the last couple of years. We have a large Mpox research agenda. I will not belabor that, but I want to show you two studies that were presented at ID Week you may have heard about. One is the PALM-07 trial, which showed the tecovirimat, which we used, TPOXX, during the outbreak here, was not effective in preventing lesion development or symptoms or mortality in adults and children of the DRC. What I would say about this, so this was disappointing, but the mortality in this study was so much lower than what we had on record for people in the usual setting. What it really showed you was that high quality supportive care can be almost better than any biomedical intervention you could give. So I feel like that's something that really should be part of the headline because people, if they get to a hospital, get hydrated, get pain relief, they did much better historically than their counterparts. Of course, we're still studying tecovirimat. If you have patients, you want to go ahead and enroll them in STOMP. That's being run by the CDC right now. And we also just published data showing that the Jynneos vaccine is safe and at least immunologically effective in adolescents. So again, thinking about how do we make sure that the tools we have can get to the people who need them. A couple of words about Marburg, largely because this is so far, fingers crossed, looking like maybe a success story if you've been following it. Given the time, I won't remind you how this is transmitted, but interestingly enough, the outbreak in Rwanda is occurring in the context of what's called pupping season in bats. So it's basically bats are breeding and bearing baby bats. And so there's a lot of opportunity for turnover, probably a lot of interaction with the outside world. And bats almost certainly, of course, as they typically are with Marburg, were the source. This is also out of date already, but what it's telling you is that there have been a number of deaths, which is really tragic, but also a number of people recovered. I just heard the Rwandan Minister of Health yesterday saying that someone in the ICU had been discharged from the ICU, which is pretty big victory when you think about these hemorrhagic fever viruses. And more importantly, no cases have been exported that we are aware. And they believe that inter-country transmission has been effectively shut down. Why did that happen? Because we have worked in the last 6 weeks to get super mobilized, to get monoclonal antibody, which originated in Jim Crow's lab at Vanderbilt from a patient who was exposed to Marburg in a cave. You may remember he presented that at ID Week several years ago. It was really fascinating. And vaccine that also has its origin in our Vaccine Clinical Research Center. So they have vaccinated now, I believe, over 1,600 people in Rwanda, and they've administered many doses of the monoclonal antibody. Last thing I will just say is I want to mention one thing about perinatal HIV transmission, and I was really fortunate to see Debbie Prasad last week, again at ID Week, and this is something that is such a no-brainer that it makes me really crazy that we still have 1,600 transmission events globally. No, 160,000 transmission events globally through perinatal transmission. When we clearly have effective antiretroviral therapy, most of those transmissions are occurring in non-PEPFAR countries. So what does that say? You need to get the programs to the people. We know what to do. We know what works. And I became aware of what happened in Maryland recently, right? You have had several cases of perinatal transmission in 2022. And I show this because this happened right before I left in Alabama. We had really not seen perinatal transmission for a long time. The exact same thing happened. And so I think it just goes along with the congenital syphilis story, right? We're seeing incredibly high rates of congenital syphilis. We know how to treat syphilis. We know how to diagnose it, kind of. It's not as easy to diagnose as HIV. But we know the tools that work, and there are just so many barriers for some of our patients and some of our communities. So the implementation side of science, which you all have worked on in concert with us in the program that has been led by Henry and Elena, I mean, these are really important aspects. For a long time, the exact same thing happened. And so I think it just goes along with the congenital syphilis story, right? We're seeing incredibly high rates of congenital syphilis. We know how to treat syphilis. We know how to diagnose it, kind of. It's not as easy to diagnose as HIV. But we know the tools that work, and there are just so many barriers for some of our patients and some of our communities. So the implementation side of science, which you all have worked on in concert with us in the program that has been led by Henry and Elena, I mean, these are really important aspects of what we do. The bench is fantastic. It's cool. I love it. But if we can't get the interventions to people, then we really can't do what we need to do. So I'll stop there. And again, I want to thank you for your long history of contributing to all of this work and for the future opportunities that I know you're going to create and to make things so much better. So thank you very much.